Well, good evening, everyone. I'll just let the last people take their seats up in the top. And we are absolutely delighted to welcome Professor Paul Nurse back to Wolfson to give the Haldane Lecture again. A lecture which commemorates the Haldanes, a family of eminent scientists who live on the site that is now Wolfson College. As I hope everyone will know, just looking at seats, there are some seats down the front. As everyone I hope will know, Paul is the Director and Chief Executive of the Francis Crick Institute, an independent charity established to be a UK flagship for discovery research in biomedicine. They describe their mission as discovery without boundaries, with no limit on the direction our research takes. It was J.S. Haldane's brother Richard Lord Haldane who in 1918 set out what has since become known as the Haldane Principle, that decisions on individual research proposals are best taken by the researchers themselves, which seems an extremely sound principle and not unlike discovery without boundaries. Paul undertook his postdoctoral research in the lab of Murdoch-Mitchison at the University of Edinburgh between 1973 and 1979, and during that period he worked on fission yeast, identifying a key gene and then its corresponding gene in humans. I should just mention that Murdoch-Mitchison, as we've been discussing, was the son of the Labour politician Dick Mitchison and his wife, the writer Naomi, who was one of the most famous of the Haldanes. The biologist J.B.S. Haldane was therefore his uncle, and the physiologist John Scott Haldane, who tinkered in his own gas lab right where the family accommodation is just over the corner there, was his maternal grandfather. In 1979, Paul set up his own lab at Sussex University, and then in 1984 he joined the Imperial Cancer Research Fund. In 1988 he came to Oxford to chair the Department of Microbiology, and then returned to the Cancer Research Fund as Director of Research in 1993, and then became its Director General. In 2001, Paul, with two other collaborators, was awarded the Nobel Prize in Medicine for that work on genes. In 2003, he became president of Rockefeller University in New York City, where he continued his research. And in 2010, he succeeded astrophysicist Martin Rees for a 5-year term as president of the Royal Society. And in the same year, I think, he also became the first director and chief executive of what is now the Francis Crick Institute. Tonight, Paul will consider the deceptively simple-sounding question, what is life?
Well, thank you for that introduction, Tim. When you read out all those jobs, it just sounds as if I can't keep a job, really, and have to move on. It's a pleasure to be back here. I have indeed given a Haldane Lecture now a decade or thereabout ago. And actually, Anna, my wife, Anne is in the audience at the back somewhere. We live in the Woodstock Road, so it isn't actually very far, though, of course, I work in London. You are quite right. It's a deceptively simple question, what is life? But it is one of the most fundamental questions in biology. Easy to ask, but as you will gradually learn, not so easy to answer. But I will have a go at it. I'm not copping out. I will have a go at it. Let me start with the extraordinary diversity of life on our planet. Because all around us, there's life, bacteria, fungi, plants, animals, including ourselves, of course, human beings. And what I want to really talk about is, what is it that links all these different life forms together? Well, it's a long time ago But I think I first started thinking about these sorts of problems when I was 12 or 13 years of age and I saw this yellow brimstone butterfly. Well, not this particular yellow brimstone butterfly, but a yellow brimstone butterfly. Early spring, I was sitting in the garden when this quivering yellow butterfly flew over the hedge, settled, got disturbed, flew up, flew around, and flew over the opposite fence. Now, that intricate, perfectly formed butterfly made me think, because it was both utterly different to me and yet somehow really rather familiar too. Because like me, it was obviously alive. It could move, it could sense, it could respond. It operated with purpose. And I started wondering then, what does it actually really mean to be alive? And that is, of course, the question, what is life? And it's a question I've thought about really most of my working life. I'm a research biologist, and I always come back to that question. However, of course, I'm not the first to ask this question. For example, this is the title page of a book, one of several books actually with the same title page. This one was written in 1944 by the famous physicist, high-energy physicist, Erwin Schrödinger. And his book was very influential, including for me. I first read it over 50 years ago. And I want to consider the same question as Schrödinger asked, but the approach I've decided to use for this is to identify what are, for me, the most important ideas in biology, 5 of them. I want to describe where they came from very briefly, what they are, and then I want to bring together those 5 ideas to develop principles which are fundamental to life and therefore understand what life is. So that's what you're going to hear about now. First of these ideas is this one. It's the concept of the cell. I call it biology's atom. The cell is the basic structural unit of life. It's also the basic functional unit of life. That's why I call it life's atom. They were first discovered and observed by Robert Hooke in the city of Oxford. And if you go into the high street, you will see a plaque on the wall there celebrating his discovery of cells in the 1660s. And what he did is he took a razor and cut thin slices off a piece of cork, which is a dry plant, and using a microscope like that, an early microscope, which had been invented about 40 years previously in the Netherlands, and he observed and drew the picture, it's published in a book called Micrographia, which looked, as you can see there, is a lot of boxes. And he called those boxes cells, after the Latin word cella, for small cubicle. And I put in a scanning electron micrograph on the right to show that we haven't really advanced very much in the succeeding over 300 years. So this is the first picture of a cell. Now, a few years later, a Dutch draper working in Delft called Anthony van Leeuwenhoek scraped between his teeth and put what he scraped out between his teeth under a microscope and discovered single-celled life, bacteria. This is a drawing he sent to the Royal Society in London. I've seen it. He actually sent about 1,000 letters to it was at the birth of modern science, basically. And I find this really rather charming image because if you look, there's various bacteria here, but if you look at the figure C to D, you see it's done a loop under the microscope. Because if you actually look under a microscope at microbes, they're moving around all the time, and I just find it charming. You see, it's the loop going from B to C to D. Now, it turns out he was a close neighbour in Delft to the painter Vermeer. And Vermeer, very unusually, he only had about 40 paintings that survived to today. It's going to be a massive exhibition, by the way, in Amsterdam, which I'm pleased to say I have tickets for, in a couple of months' time. But he painted 2 pictures which were of a scientist. Now, scientists were not normally painted in the 17th century. This is one of them. It's the one that's in the Louvre. And I like to think that this is Leeuwenhoek, because he lived near Leeuwenhoek. But I have to tell you, there isn't any good evidence for that. None whatsoever. Although there is a connection between them, because when Vermeer died, he died in his early 40s, Leeuwenhoek was actually appointed the executor of his will. So it's not completely crazy, but there's no good evidence. But I like to think it's true. And it allows me to show you a very nice picture as well with this. Now, over the next 2 centuries, it became really clear that all living organisms consist either of a single cell, that's single-celled life, which is what Leeuwenhoek had discovered, or a collection of cells, such as our cells. And this conclusion was nicely summarized by the German zoologist. There were many others who contributed to this. Theodor Schwann, who wrote in 1839 in German, but translated here, that all organisms are composed of essentially like parts, namely of cells. And that's why I said the cell is the basic structural unit of life. But it is also, cells are also the basic functional life. And that was nicely summed up by yet another German scientist, the first pathologist, really, Rudolf Virchow, who in 1858, so about 25 years later, wrote, and you see it here, every animal appears as a sum of vital units, each of which bears in itself the complete characteristics of life, the functions of life. So there you have the concept of the cell. He also said, by the way, that all cells come from cells. He wasn't the first to say that, but he's the one who's most often quoted in Latin, omnis cellula e cellula. And what this means, of course, is that cells arise through the division of pre-existing cells. I'm going to return to that in a rather interesting way towards the end of my talk. And why that was important was to push back on vitalism, whereby cells just arose in a sort of in mud or dirty water or whatever. This meant cells arose from pre-existing cells. So cells are interesting. We don't actually, I think, talk about them quite enough. But if I fail to convince you that they're interesting, let me show you this picture, which is a picture of an egg with lots of sperm trying to fertilize it. And then I can remind every one of you in this room that you were all once a single cell, just like this. And if your interest is still not piqued, there is a door at the back of the lecture theatre. Now, cells are considered to be the simplest entities which unambiguously exhibit the characteristics of life. I say unambiguously because I will have to talk about viruses a bit later, which are a strange beast in this discussion. And what this means, therefore, is that understanding cells is key, actually, to understanding life, because you see it in its simplest form looking at a cell. So they will figure quite prominently in what I have to say. So what is a cell? It's a bounded, this is a picture, it's coloured artificially, just to show the various components in a cell. It's a modern photograph, or fairly modern. Cells are bounded physical entities, remember that term because I shall return to it in a minute or two, separated from their environment, surrounded by a lipid, semi-permeable membrane, so separated from their surroundings, but in communication with their surroundings. Why am I emphasizing that separation? It's because the separation allows order to be built up inside cells at the expense of increasing disorder outside the cells, because they're separated. And in this way, life does not contravene the second law of thermodynamics. And that's important, which of course states that the universe as a whole is moving towards disorder, because there's local order possible within the bounded cell. And this law, the second law of thermodynamics, often troubles physicists who think about life, including Schrödinger, as you can see in this quote from his book, the one I mentioned to you. He states, I'll read it from here, and he noted an organism's astonishing gift of controlling a stream of order on itself and thus escaping the decay into atomic chaos. What was struck by him was the successive cell divisions producing cells with order, which was maintained. And he could, as a physicist, he thought he was a little uncomfortable about that. But there isn't a problem. We get away with it because it's separated from the rest of the universe. But his second quote, which I've stolen from that book, is an introduction to the second idea, which I want to talk about, which is heredity, because he also proposed that that cells and the chromosomes within them must contain some sort of code script which governs the individual's future development and functioning. So that leads me on from the cell as the first idea to the second idea, which is the idea of the gene, which can explain that mysterious phenomenon of inheritance. That is, the gene is the basis of heredity. Now, that idea had its origins with the work of the Augustinian monk, Gregor Mendel, abbot of Brno University, who published his work in 1866. Here he is. This is Gregor. I was in Brno. It was his 200th anniversary of his birth this year, and I went and gave a talk at the 200th celebrations there. Met all the present monks. There's a total of 6 of them left in the monastery. Now, he was an extraordinary person. He tackled the problem of inheritance by crossing pea plants, which had different characteristics, and analysing the outcomes of those crosses in the progeny formed by those crosses. Now, this was quite a popular research subject in the middle of the 19th century because plant scientists, botanists, were trying to understand heredity for crop improvement. And so they were crossing different plants to try and see how they could improve them. And the more intellectual of them were trying to work out what that meant for heredity. And the truth of it was that it was a complete mess. Okay, and Mendel made some sense of it, and I'll briefly say why I think that was. I visited actually his garden, and this is a picture from 1981. I went in the middle of the Cold War, and quite difficult to get there, I have to say. It was in winter, and this was his garden. Actually quite a big garden, if you think about it, for a monastery. And what I discovered, and I hadn't known before, was that adjacent to it was a greenhouse of the same size. And the abbot before him, because he became abbot, funded his research, building big greenhouses, big gardens, giving him a research assistant. And in other words, the monastery was a research institute. I mean, it's not how we imagine it today, but that's how it was. And that was largely because the Augustinian monks were a teaching order, partly, and they had linked research with teaching. Now, Mendel wasn't very good with exams. He was trained as a physicist so he could become a physics teacher, and he kept failing the exams in Vienna, University of Vienna. And he wasn't good at that at all. I'm quite pleased about that because I wasn't very good at exams either, it has to be said. But he did bring to the biological problem a sort of physicist thinking, and in particular, he was quantitative in his thinking. In other words, this was a time, and we're going to hear about Darwin a little later, of the great naturalists who described what they saw, and sometimes brilliantly so, making conclusions. But they weren't very quantitative. And what he did is he crossed and there's various examples of the different varieties of the pea. So you had tall ones and short ones, different coloured flowers, different coloured seeds, and so on. And he spent a lot of time preparing which strains he would cross so that he knew he could clearly identify what that we now call the phenotype is. That is what it looked like. And therefore, he would be able to count what the progeny were, what type and so on that they were. And when he went through all of this, he began to realize that there were simple ratios in the progeny that were being produced. The most famous one, which maybe you learnt at school, is a 3-to-1 ratio. When you cross two different varieties and then you cross those, out you get is 3-to-1. Now, I'm not going to explain all of that. I could, but it will take up, I've got 3 more ideas to do. So just believe me that you can make sense of it. And this eventually led to what is called Mendelism. And I say eventually led because actually Mendel didn't quite get it right, really. He almost got it right, but it required a little more work from others to actually get it as clear as to the idea of Mendelism. And that is the idea that heredity was based on pairs of indivisible particles. That's why you could get nice ratios out of it. Particles or factors, one of which is inherited from one of the germ cells and the other from another of the germ cells, which become joined together during fertilization. That's the pollen and ovules in plants, sperm and eggs in animals. And these factors were subsequently found to be, and this, by the way, was revealed by him counting, because anybody would have revealed them, but he counted it. In fact, I discovered reading one of Charles Darwin's books, he also did crosses, in his case snapdragons. He also observed a 3-to-1 ratio, and he describes it, but they moved on. He just moved on. He didn't think about it, and I think it was the quantitative and somewhat mathematical thinking that led to Mendel thinking about it. Now, these factors were subsequently found to be linked to the chromosomes, which were discovered about 20 to 30 years after Mendel's death. And here, what you see here is a picture from the late 19th century of onion root tip cells, squashed under a microscope. And you'll be able to see, of course, the chromosomes separating here. By the way, I first saw cells and first saw chromosomes from an onion tip when I was about 14 at school. So this I can confirm this is correct. I can confirm it's correct. Now, then people began to start taking this seriously, and I think actually there's something interesting there, because Mendel's analysis was ignored. Nobody took much notice of it until 35 years later, when 3 other geneticists, early geneticists, repeated the same observations. The first one ignored Mendel altogether. The second one, I think, piqued that he wasn't the first to rediscover, brought a lot of attention to what Mendel had done, so he could downgrade, presumably, the work of the first one. And then the third tried to sort of find a middle way. But what I think is interesting about it is that Mendel's analysis was abstract. It was an abstraction, whereas seeing the chromosomes behaving like it is a concrete observation. And it was the linkage of an abstract thought to a concrete observation that actually gave this idea so much more energy. Okay, so these Mendelian hereditary factors are what we call genes. In the 1940s, Oswald Avery, who was at Rockefeller University in New York, where I was president, showed that genes were made of deoxyribonucleic acid, made of DNA. His colleagues there criticized this work for decades, actually, but he was absolutely right. And I remember when I went there in the early 2000s, this little old man came up to me and introduced himself as McCarthy, Professor McCarthy. And I said, "You're not the Professor McCarthy who worked with Oswald, with Avery." And he said, "Yes, I am the Professor McCarthy," as indeed he was. Okay, so the structure of DNA as a double helix was established 10 years after Avery in London from the work of Rosalind Franklin, Ryan Gosling and Maurice Wilkins, who did the X-ray crystallography mostly, and in Cambridge, who interpreted that data by Francis Crick and Jim Watson. And Crick and Watson famously argued that this double helical structure, and we're going to talk about this later, can explain heredity through the linear sequence of bases here making up DNA being precisely copied. And so what you can now see is within the nucleus are the chromosomes. The chromosomes are made of DNA. The DNA has this double helical structure, and that is sort of at the center of understanding heredity. Now, you're going to see this slide twice, actually, because this is just to show to you how it can be precisely copied. Because what we have here are, if you can imagine the double helix as a twisted ladder, and you have rungs across from one side to another, but it's easier just to imagine it now as a ladder, then the structure of DNA chemically is that on each side of that ladder, the rung is made up of two complementing bases, an adenine, which complements to thymine, a guanine that complements to cytosine, and they can form a nice bond with the rung, whereas an adenine can't do that with guanine because they don't meet properly, okay? And so once you've understood that principle, which is what Watson and Crick did, if you split it, then you can very precisely make an exact copy because it forms a template for the copying. And obviously, since this is the way DNA is copied during cell reproduction, it can account for heredity. It's a very beautiful idea. It explains Schrödinger's code script. It explains the permanence of the gene between generations, because the DNA acts as a code script, which is precisely copied every time a cell divides. Also, and I'll perhaps remark on it, the information that's stored here is sort of turned in the middle of the molecule and sort of actually protected there by the outside. Crick went on to propose what he called the central dogma, which probably says something about his intellectual arrogance, of course, but where the information encoded in the DNA is transcribed into a message, messenger RNA, and that in turn is translated into a different platform, essentially, of a sequence of amino acids that make the proteins. And we're going to talk about that in a minute, because the proteins turn the inert informational store into chemical action. Now, these discoveries were all made in the second half of the 20th century. They laid the foundations for molecular biology and much to do with applications in health, medicine, and agriculture since then. And they are critical, which is why I'm emphasizing it, for understanding life. Let me say a little bit more about it. Central to this is the genetic code. It's written in this four-letter code, four bases: adenine, thymine, guanine, cytosine. Now, this way of storing information is linear, and it's just like reading sentences in a book or listening to me speaking to you. I'm speaking in a linear form. Its speech is in a linear form, or the chain of bytes in a computer. They are all digital, linear, information linear storage devices. And I think to put this in perspective, that means that linear digital storage of information was invented by life about 3 billion years before the computer age. About 3 billion years before, just to keep us humble, you know. Now, the nature and function of the gene introduces the next two ideas: life as chemistry, which I'm going to talk about next, and life as information. Now, the functioning of living organisms based on chemical activity and physical forces is an important idea. It can be traced back to French chemists. Initially, the work by Lavoisier, just before the French Revolution, who was basically the main founding father of chemistry, in actual fact, unfortunately lost his head in the French Revolution because he was also a tax collector. He was very liberal thinking. But he lost his head, so that stopped his research, naturally. But he also, very famously, when he was tried, it was a kangaroo court, it said, the head of the court said, the revolution has no need for savants, for wise people, or chemists. I always say, when you're a scientist, it can be dangerous to interact with the politicians, of course. But it was after that initial work by Lavoisier, it was Louis Pasteur, this is Louis Pasteur, heroic picture of him, who carried out experiments on sugar beet in the north of France, which led to a better formation of this idea that life is chemistry. Now, what Pasteur was doing was he was trying to help the local industrialists who did sugar beet fermentation. So sugar beet, as I'm sure you know, has got lots of sugar. And if you can ferment it, you can turn that sugar into alcohol. But sometimes those fermentations fail, and they make, instead of alcohol, they make acid. And the industrialist came to him and asked, can you help us? You know, this was an applied project here, of course. And what he did is he simply looked at everything under a microscope and so on, and discovered that when you got a successful fermentation that made alcohol, you had yeast in it. Okay? When you got an unsuccessful one which made acid, you couldn't see any yeast, and probably bacteria were there. So he helped the industrialist by saying, just put some yeast in there and you'll get alcohol. Okay? Very useful. But he concluded more generally that therefore that fermentation was a physiological act which yielded products for the cell, the yeast cell. And therefore, chemical reactions are an expression of the life of a cell. This is around the 1860s, 1870s. That is essentially saying life is chemistry. But it isn't any ordinary chemistry. It is a very, very elaborate and special chemistry. Now, carbon polymers are crucial for this. We've already met carbon polymers that make up the nucleic acid. I haven't told that to you, but that's what they are, of DNA and RNA. But now I want to talk a bit about proteins, because the substances from yeast that Pasteur postulated made the alcohol are proteins that act as enzymes, and those enzymes catalyze chemical reactions. Now, proteins are polymers of amino acids. I briefly mentioned that. They're based mainly on carbon and nitrogen, which is why I said they're carbon polymers, which form the backbone to that polymer. And there are the amino acids also, as well as having the atoms that form the backbone that you can see here, all carbon and nitrogen going here. They have, the different amino acids have different side chains, chemical side chains. There's 20 of them in in proteins from living things. And these different side chains give different chemistries as a consequence of it. So some of these are positively charged, others are negatively charged, some are hydrophobic, they don't like water, some of them are small, some of them are big, okay? And the result of this is that proteins can produce, firstly, elaborate structures because of all this different chemistry. They can wind up in different ways. And secondly, they're chemically diverse. Whereas DNA and RNA actually is chemically dull because all it does is store information. This is a really important concept. It's not actually stressed enough. The information is dull. It produces proteins which are more unstable because they're chemically active, which can carry out chemical work. Now, this is another look at that. This is a bit more of a structure. And here I haven't emphasized the side chains. They're just here. This is just to emphasize the backbone. But the side chains are there as well. And a consequence of this is that when you have a polymer, then the polymer can fold up in different ways because it links to different parts of the protein. Because there's different, I mean, simply, just to think of an electrical one, if you've got a positive charged amino acid and a negative one, they will form a bond. And so you can now wrap the protein up into, here it's in 2 dimensions, here it's in 3 dimensions. So look at this molecule, it's colored to show there's different chemistries, because what this produces is elaborate structures which also have elaborately different chemistries potential in different parts of the structure. And that's really what I wanted to emphasize over that. And that accounts for why there's a huge diversity of chemical reactions that these enzymes can carry out, because all of these are different in different sorts of ways. It's a magnificent chemistry. They act as chemical machines. The machines produce the huge range of chemicals that are required for living beings to function. They make new molecules, they can break them down, recycle them. They can generate large molecular assemblies. They can synthesize these polymers of nucleic acid and protein, lipid, carbohydrate. They can do physical work. They can make motors. Motors that can carry cargoes around the cell on protein-based railway lines, essentially. It's amazing stuff. They can act in hybrid ways, combining chemistry and physics, capturing energy from sunlight, from light, or breaking down sugars to produce energy-rich chemicals that can drive the activities being carried out in the cell. And all this, what this all means is there are thousands of chemical reactions being carried out simultaneously and close to each other in tiny cells, which are only 10 microns across, 10 micrometers. And this is why I said a few sentences ago, this is extraordinary and special chemistry, and it is the basis of the functioning of living things. And it is incredibly elaborate. And what you see here is this is a map of some of the chemical reactions, the metabolism that's going on in cells. For those of you who may remember the Krebs cycle, that's here. There are a couple of hundred of dots on this. There are thousands of dots. And all I'm trying to communicate to you is all these reactions are going on within 10 microns of each other. And that is why it is completely extraordinary. Now, how is that possible? Because each of these different reactions requires different chemical conditions. That's why and so this requires them to be separated one from another. And this is achieved by compartmentation. It can be brought about by the folds of those proteins, because they can make little chemical sort of safe spaces, okay? It can be brought about by the complexes of different enzymes acting together. It can be brought about by membrane-bounded structures within cells, or by other chemicals, other chemical assemblies called phase separation, which have been discovered in the last decade or two, which mean you can isolate different parts of these chemistries. And that's actually what you're looking at when you see this cell. It's not the way it's normally talked about. You know, it looks static. It is not. These different structures here are actually providing large numbers of different chemical microenvironments which allow the hugely elaborate chemistry of life to take place. And I show you this just as a more sort of schematic thing to sort of emphasize the same thing. So that's extraordinary, I think you agree. But remember, and this is now going to be a transition to the next idea, life as information. For this to work, it's got to be coordinated. It's got to be working together as a whole. And that requires communication between these different chemical microenvironments. They have actually to be separate, but they have to communicate. And communication is the transfer of information. That's what communication basically is. And that's going to lead to the next idea, life as information. And that touches upon the fact that life is a complex system, it's controlled, it acts with purpose. Now, I hope that was okay, the chemistry. These are the two difficult bits, actually, chemistry and information. But we got through half of it, so you're prepared for the next half, okay? Now, life as a complex system, it sort of sounds obvious that it has to be. But I did a little work to try and find who was the first person to start saying that. And I discovered it was the philosopher Immanuel Kant. And sometimes you're really surprised about what you discover, but these were polymaths, 200 years ago. And he wrote a book on moral philosophy and has about 20 pages on the complexity of life and arguing that it's a complex system. It's true. It's quite extraordinary. First came across it at the University of Sussex. You said I was there. Sort of thing they do in Sussex, or did then, I should say. And these complex systems of life require the management of information. And I'm going to give you two examples of this. And for the first one, I'm going to return back to the structure of DNA, which is the iconic one. Now, I explained it here, but let's just now restate it in terms of information particularly. Now, the structure of DNA is quite beautiful. It is iconic. Although I do get tired in any, you go to any life sciences department or institute and there's a damn double helix somewhere in it. I mean, we could have more imagination. When I put the crick up, I said, I don't want any double helices in it, okay? I don't want any, okay? But although it's quite beautiful, its real beauty lies in the fact that it only makes sense, the structure of DNA, when you recognize it for something I've already said, that it is a digital information storage device. That's what makes biological sense of the chemical structure. The chemical structure is uninteresting until you realize it's a digital information storage device written in these 4 letters. And I said, it's protected inside the molecule. But as I've already hinted at, this information within the cell can be turned into chemical action because that information can be translated into the proteins that give rise to all that different chemistry. And for me, this is an absolutely wonderful characteristic of life built on carbon-based polymers. The ability to encode information in these linear structures, and secondly, for that information to be translated into elaborate chemical structures that are active enough to do chemical and physical work in 3 dimensions. And that is central to how life works. Now, the second example, and it has its origins in the structure of DNA, but you have to put everything together. The second example of information I want to tell you about is actually to do with regulation. And this is really important in understanding living things. Now, I don't know if you're like me. I understand most machines built before 1900 and increasingly no machines built later. It's like looking in a car now. You used to be able to fiddle with it to get it to work. Now, you can't do anything, unless it's 50 years old. And so I'm going back to a steam engine I discovered in a boat in New Zealand. And this is an example of a governor developed by James Watt on how to regulate steam engines. Now, you can understand this, you see. This is connected to the rotating spindle driven by the steam engine. And as this rotates, the balls go out, okay? Centrifugal force. That it turns off a valve, it cuts off the steam going into the engine, right? So then the engine slows down until the balls now come back in again and the reverse goes on. So this is actually a homeostatic mechanism that leads to homeostasis. It's a negative feedback loop. And that maintains a constant speed of the engine. Without having somebody driving it, it just happens naturally. And of course, that's what happens in cells. This is a metabolic pathway. I've just described to you a negative feedback loop. So what you've got to imagine is a chemical A turns to B turns to C, and C inhibits the A to B. So if now you get too much C, it can turn off A to B, and the amount of C drops. Okay? Negative feedback loop brings homeostasis. Positive feedback loop, let's look at that. Complete reverse. As C builds up, you promote A to B. So this becomes an irreversible switch. Once you start going this way, you have to keep going. There's no way of actually going backwards. And these are the simplest sorts of controls you see in living cells or living organisms. But what we see is combinations of these and other controls that can maintain homeostasis, generate switches, as I've described, make timers or togglers or oscillators, or translating chemistry into informational modules. Because now we're explaining how the chemistry, we're not just sort of fiddling around with chemical reactions, we're now talking about the management of information and maintenance of structures. And these can be linked together to make even more elaborate routines of informational management. Now, this is a metaphor for all of this. This is, I think, a radio, and some wit, I got this out of a journal, some wit, these are all oncogenes, they cause cancer or switch cancer off, and somebody sort of identified these different components and put all this together. And this is, if you like, the hardware of of a digital radio here being thought of as being a metaphor for the information management in living cells. Actually, for those of you who are particularly interested in this, cells are cleverer than this. This is hardware, it's hardwired. Actually, the way this works in living cells is through liquid solutions. So you can rewire your hardware. So Dennis Bray, the systems biologist, called this wetware in comparison to hardware. It's a nicer metaphor. But it increases the versatility of control modules if you can rewire them. This, by the way, we haven't yet explored enough in biology, but it's going to be important. Okay, the management of life's chemistry by information allows eventually the behavior of that complex system to act with overall purpose. I mean, that's how it emerges from this in some way. And therefore, you can grow a cell, you can reproduce a cell, you can make a butterfly, you can make our cells by putting all of this together. I don't know how it all works, but that's the principles by which it works. Now, I said it that way, purposeful behaviors. I'm deliberately being slightly provocative because for centuries, the generation of purposeful behaviors was thought to require a designer, somebody who had designed it, a divine creator. And it took evolution by natural selection to change that. And this is my final idea, the fifth idea. If I'm going on a bit, we're getting towards the end, okay? So deep breaths, we're nearly there. Not quite, but we're nearly there. So evolution by natural selection, how do living things come about? Now, this is probably the most beautiful idea in biology. It's mainly due, but not only due, to Charles Darwin, and it has two parts. The first is that life evolves over time, and the second is that a major mechanism of it is natural selection. Now, Charles was not the first to suggest that life evolves. In the century before him, Jean-Baptiste Lamarck in France, and Charles' own grandfather, Erasmus Darwin, both wrote extensively on the fact that life evolved. I like Erasmus. If you don't come across him, he was a much more interesting person as a person than Charles. He was colourful. He was a scientist, a doctor, and a poet. And most of his research was written in poems. Really is written in poems. I have three of his first editions of his book from the 18th century. And it's all there in this mad, free verse, okay? He was a Republican. He refused to be the doctor of George III. He was asked several times. He was a good doctor, by the way, not because he could cure people, because no doctors could cure people, really, at that time, but he at least was very good at telling you whether you were likely to die or not, and that was very helpful in that society. He was a proponent of women's education and wrote a book for his two daughters to set up a school for girls. And he had a terrible stammer, but was a great conversationalist. He lived in Derby and just north of Birmingham. Nowhere near a university. Nowhere near a university, has to be said. And he argued for the evolution of life. And he had on his coach a motto which said, all life from mollusks. Because a mollusk, like a snail, is a sort of formless thing. And he was saying that's from which life can come. This wasn't very popular. He then did that when he was in Lichfield. He lived in the Cathedral Close, and the dean wasn't very keen on this, it has to be said. And he had to paint it out because he was losing all his rich patients because they didn't like it. Now, he argued, therefore, for the evolution of life. Charles did. But the implication of that is that there is a tree of life, which I want to bring to your attention. That is, all living organisms are related, and if life only arose and survived once, as seems probable given the conserved chemistry and coding I've been talking about, then that means all life on Earth is related to each other. We're all related. And this connection of life was captured in the only diagram in Darwin's... by the way, this I should have shown you this when I was talking about Erasmus. That's Erasmus over the left, okay? He liked his food and wine too, by the way. And there's a rather serious Charles on the right. This is the only illustration in Charles' Origin of Species, and it's the first tree of life, which we're now very familiar with. Notice this. Did it arise more than once? He was keeping his options open. This implies that it might not have done. I argue, given the similarity in chemistry, that perhaps it did come only once, or survived only once. He came to these ideas through the famous trip in the Beagle, 5 years on a boat doing surveys for the Royal Navy, and he proposed natural selection. Now, what is natural selection? Well, it's a consequence of populations of living organisms, same species, and it's a consequence of them exhibiting variations which are caused by inheritable genetic changes. And then these can be caused by mistakes when the DNA is copied or damaged or mixed up in different sorts of ways. And if these variants influence characteristics that can make these organisms less successful, then they just die out. But if more successful, so that the organism is better adapted, then the better adapted organism will reproduce more, and therefore over time that particular variant will spread through the population. It's a bit more complicated than that, but that's roughly what it is. And over time, such genetic variations accumulate and lead to changes, and eventually to sufficient changes that the new population that is made can no longer breed with the original population, which is why his book was called The Origin of Species, because that's the definition. He came to this view in part by the study of finches on the Galapagos Islands. He didn't do it while he was there, it was when he came back to London. And I've stolen this picture to show these are the different beaks, and these are man-made pliers, okay? And the beaks are specialized in different ways. This one cracks nuts, heavy thing. This one gets insects under the bark of trees. And we have produced different pliers for different jobs, okay? But the difference is, of course, that this arises by natural selection and this is intelligently designed. But this is the reason why many people in certain parts of the world have real trouble with this, because they always think it has to be designed, because they're not happy with this explanation. Now, I'm now going to sort of pull a couple of things together here, because for natural selection to take place, you need three things. One, life has to reproduce, okay, obviously. Two, it has to be controlled by a heredity system. Three, that needs to exhibit variability. And I want to show you how that works by building on two of the ideas I've already talked about, the idea of the cell and the idea of the gene. Because here we have a cell, here we have genes, and the genes determine how those cells work, right? That's where we start. Now, imagine in this simple organism that when you have a brown coat, you taste good to a predator. But if you have a red coat, you don't, okay? Now imagine what happens here, that by mutation or some mistake in replication, you alter a gene which is important for determining whether it's this brown coat or this red coat. And now we have a lineage of cells with a red coat, okay? And the yellow ones get eaten by the predator, and the red ones don't. So the red ones will replace the yellow ones. And that means that by combining the idea of the gene and the cell and making certain assumptions, then you can understand evolution by natural selection. Now, this is why this is such a profoundly important idea, simple as it is. So much so that the 20th century geneticist Hermann Muller, who worked in New York in the 50s and 60s, Russian origins actually, he used that to define life. And that, I'm going to introduce that now because I'm going to use it before. Use it again a bit later. He said that living things can be defined because they have properties which allow them to undergo natural selection and therefore to evolve. It's a little bit of a cop-out because it doesn't sort of explain, how life works. But without evolution by natural selection that can produce organisms that are better adapted and therefore function better, you can't imagine it. You can't imagine how life could go on. Now I'm going to turn to that in a moment, but first of all I want to say goodbye to Darwin. Okay, mainly because of this great sentence, the last sentence in his book, which is, whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. Why can I not write sentences like that? And why, if I could, will they be edited out by the editor or the reviewers? I mean, you can still read this stuff 150 years later and it says something to you, You can't read anything I write after 5 years, it's completely redundant. So here he is in his late 70s, and notice what he was doing here, because he was a good salesperson, really, because he connects this to the law of gravity. The law of gravity, of course, is central to physics, comes from Isaac Newton, and he's saying biology is like physics. We have laws that can explain natural phenomenon like life, just like physics do. Now, before I finally finish with defining life, I want to consider the difficult category of viruses, okay? Because that's what people always ask. And then we're near the end. Now, viruses. This is a virus. It's actually of a bacterium. It's a bacteriophage. They have a genome based on DNA or RNA. They undergo natural selection, as we've seen with all the COVID variants. Thus, they pass Muller's test. But the problem with them is that they can only reproduce and function when they're inside the cells of other host living organisms, which is why the question is, are they alive or are they not alive? And they do that by hijacking the cell's molecular machinery. And that means the virus is completely dependent upon another living entity. So that's the conundrum. Now, there's various ways you can deal with it. Sometimes I say, well, you could say a virus is alive when it's inside the cell and is dead, chemically inert, when it's outside. But I don't like that. And so I'm going to give you a different view of this. So in thinking about whether viruses are truly alive, it's very important to remember that other forms of life are also, to a greater or lesser extent, dependent upon other living things. And that's what we forget when people say about viruses. The many parasites that live on or inside the cells or bodies of animals, plants, or fungi are examples of this. But we are all dependent upon it in different ways. What do you eat? You eat other animals and plants. There are certain amino acids we cannot make chemically. We have to get them from the amino acids made, for example, by bacteria. So there is, in fact, in life on this planet, an extraordinary, diverse, interacting network of dependencies. We cannot view ourselves separately from the rest of the living universe around us. And we are dependent to varying degrees, we being an example of life, on other living things. Now, once you've accepted that, then the virus no longer looks quite so different. Because what it means is there's a graded spectrum of living organisms, from the viruses through plants, which are a bit more independent, with a wide range in between, which includes our cells. These different life forms have varied dependencies on other life forms. In the case of the virus, dependency is strong. In others, it's weak. But these are all alive because they evolved by natural selection. And you can get out of the conundrum if you think of it in this sort of way. And this means that life is fundamentally connected, both through being related as a result of evolution, the tree of life, and also through deep interactive relationships and interdependencies. Two very important concepts. Okay, so now I'm at the point where I'm going to offer you some principles in thinking about life. I should warn you, it does not lead to a neat dictionary-like definition, which is why it's so difficult. Okay? 4 slides. The first set of principles. Living things are independent, bounded physical entities, you remember that? They're separated from their environment, but in communication. Therefore, they can produce order and organization. The basic unit of life, at least on Earth, is the cell, and all life is either a single cell or an aggregate of cells. Second, the central principle for defining life, I argue, is Muller's. Life forms are entities that can undergo evolution by natural selection, and by doing so, they acquire purpose. They are entities that build, maintain, and reproduce themselves. They act as a whole. Living things can evolve because they have the attributes of reproduction, a hereditary system, and one that has variability. Life forms are chemical and physical and informational machines which construct their own metabolism, which is used to do those things, to build, maintain themselves, to grow and reproduce. And fourthly, a bit less of a principle, but important to mention, life on Earth is based on polymer chemistry, carbon polymer chemistry, that produce linear information storage devices and protein-based chemical machines, integration of which requires the management of information. Inputs are gathered from within the cell and outside the cell, processed and stored, and used to instruct cells and organisms to maintain themselves, to grow, and to reproduce. And all living things are related by common descent and closely interconnected. So that's my definition. A few more sentences. Now, should we ever encounter life, I told you it would be long. Should we ever encounter life elsewhere in the universe, I think it will be based on rather similar principles. The details may be different. It could be different chemistries. It has to be said, when NASA looks for life, they always assume it's the same sort of chemistry, but it could be different chemistries. However, I think for it to readily serve the requirement for long-term information storage and chemical activity, it is likely that it will be built on polymers that can store information and, by various chemical tricks, can also carry out the chemistry of life. They also, of course, have to be able to gather energy, either geothermal within inside a planet or light from a star. However, I want to end by returning to our own planet, because this is the only corner of the universe where we know for certain that life does exist. We know it. The life that we are part of here on Earth is extraordinary, it surprises us, but in spite of its bewildering diversity, and it is bewildering, we are making sense of it. And that understanding makes a fundamental contribution to our culture and to our civilization, as well as the applications of it for health and other things like agriculture. And therefore, our growing understanding of what life is has potential to improve the lot of humankind. But I want to take it just a bit further, because biology shows us that all the living organisms we know of are both related and interacting, and we are bound by a deep connectedness, therefore, to all life. Bacteria, fermenting yeast, peas, flitting yellow butterflies, let's go back to that. We are connected. And together, these, and this is, it's just to finish with a bit of technicolor. Together, all of these species are life's great survivors. They're still with us. They've gone through evolution of natural selection. They are with us. And now listen to this sentence. They are the latest descendants of a single, immeasurably vast family lineage that stretches back through an unbroken chain of cell divisions into the far reaches of deep time. I know it's technicolor, but it is actually true because of what I said earlier. We're connected by life of division. We go back millions of years, billions of years. We're connected to it in time. We're connected to everything else through relationship and interactions. Now, as far as we know, we are the only life forms who can see this deep connectivity and reflect on what it means. And I argue that this gives us human beings a special responsibility for the biosphere, for life on this planet, made up as it is by our relatives. Our relatives, some close, some more distant. So we need to care about it, we need to care for it, and to do that we need to understand it, which is really why I've written actually a book about this, which is shown here. And thank you very much for listening to me.
Maybe just one or two questions if people wish to do so. As the microphone, there's one gentleman up on the left in a red shirt, if you want to.
I'm a bit deaf, so do speak into the microphone slowly, that would be good. Hi.
Thank you for your interesting lecture. My question is about evolution. So, based on Darwin's theory, evolution happens when a structure changes. So, ever since we discovered the new science, has any evolution happened? Like, do we have any new species in the last, 1 century or so?
Yes, well, I think that I'm not an evolutionary biologist, but I mean, certainly if you look at microbes, they have different species and they have different structures. But we've also seen in our lifetime, in the lifetime of human beings, dramatic changes, for example, in domestication of species. So what we now grow as wheat looked nothing like it 10,000 years ago. It was like a grass. What we now eat as a sheep is very different from what was around 10 to 20,000 years ago. Now that is, of course, artificial selection, not natural selection, so that can go more rapidly, but it is selection. It's manipulating the genome by breeding and selection, but it's a sort of fast form of evolution.
Can I ask you to pass the microphone up just to this?
Yes, there was somebody up there, so...
I think you said that life only occurred or survived once.
Well, I should say that's what I think, but I couldn't say that for certain.
How do viruses fit into that idea?
Because viruses are built on the same fundamental principles of chemistry as, in fact, other life. They use the same genetic code, or almost identical genetic code, and they have the same structures in the polymers of DNA, RNA, and protein. So it's all remarkably similar. It's the same amino acids, the same nucleotides. And that's the main argument. I couldn't have said this 100 years ago because we didn't know that. But we now do know that. So the simplest explanation, and we've got to be careful about, because it may not be correct, but the simplest one is the one I said.
But they must have split off from cellular life at some point.
Well, I of course, haven't tackled how the hell life started. And I can tell you why I didn't, because I simply don't know. I mean, it is extremely difficult to imagine how it worked. I mean, there's decent theories around, but we're looking at a period of 3.5 billion years ago. And of course, it's difficult to know quite what was going on 3.5 billion years ago. But you're dead right. You have to make a cell to get it to work. And there's different things in there. And it's difficult to imagine how they could come about. I mean, I completely agree, if that's what you were thinking.
Sort of.
Yeah.
Thank you.
If we could just bring back the microphone to Pedro for one last question, then.
Thank you.
Well, thank you for a beautiful talk. Your definition of life has lots of bits to it. And it reminds me a bit of the Ptolemaic model of the cosmos,
Yep.
which was completely transformed and done away with once Newton came up with his laws of gravity and an understanding of dynamics and mechanics came along. Do you think something like that might happen in our understanding of life?
I don't think it will, because life is much more complicated than gravity. And I think we have to capture a number of different attributes. Now, in a sense, I've tried to distill that, because although what I ended up with 4 slides, or 3 slides really, is complicated, the emphases I've put, on polymers and the chemistry and so on, are getting close to principles. And until I started thinking about this to write a book, I hadn't thought of that sort of distillation quite like that. But it does have these different things because life is really complicated. I can't quite see how we could turn it into the analogy of. I mean, I like your analogy of the Ptolemaic system in the solar system versus, and we all understand it through gravity. Though, of course, gravity wasn't an explanation. Let's just. I mean, gravity is. You're nodding. You know what I mean. I mean, it is actually just a way of predicting, in a sense. It took Einstein to sort of turn it into something that might be real, and of course, what he turned into what might be real is a bit odd too for our minds. So, although it is a single principle, it's not one that sits comfortably with our common sense brain. I'm trying to sort of distract you a bit from the beauty of gravity. But no, I think life has got to be complicated. I do not see it undergoing a Copernican revolution.
Paul, thank you very much indeed. You set yourself a deceptively simple question, and your answers were, the presentation was deceptively simple. I felt I followed you some of the way and then lost you and then came back. But that's my limitations, certainly not yours. But thank you once again for coming back to Wolfson and giving us the Haldane Lecture.
It was a pleasure. Thank you.